one being diseases related to the humans and what the compounds are prepared for various diseases. Now, this session, we have to discuss the climate change, insect pest, and agriculture systems. Because we need food, without food we cannot survive. And what scenario nowadays we have with regard to the climate changes are there, and how this climate change influence the insect behavior, and how this insect behavior is related to the agriculture. This is altogether related with the productivity of the crop, population size increasing of the insect, and what environmental factors are there. So we will go through all such topics like that of climate change and insect pests, agriculture effects, what we have in India, and another important thing, this entomopathogenic nematodes, and how this entomopathogenic nematodes can be utilized, used for the agriculture to control the insect pests. So all these four topics we will discuss one by one. I think we are familiar with these two terms, weather and climate. Weather, what the daily changes in the environment factors are there, and climate related to this with a few long time changes. But see what global warming and climate change, these two very terminologies nowadays discussed everywhere. And all these, because so many environmental factors that influence the life are currently so many studies on the basis of such changes are going on. See, this is what temperature we have nowadays since pre-industrial era. In 2025, the temperature is 1.15 warmer than what this previous era we had, and it is expected that the global warming, the temperature will increase 1.4 to 5.8 degrees Celsius. So you can imagine if such temperature is increasing, the impact of this temperature, of course, that will be harmful for the all the life organisms. And with failure of mitigate climate changes perceived as the biggest number of risks facing the world over the coming years, like that of the water natural disasters, we are facing nowadays. See this cold region still today in our north region, there were snowfalls and temperature is just decreased down, and suddenly the temperature is increasing. So of course such fluctuations are there, and that affect the life. If you see the agriculture, and the crop health is increased threatened by the climate change based on insufficient response of agriculture practices to these states. Losses of several cereal, rice, wheat, maize, and other tuber crops like potato and sweet potatoes directly impact food security and nutrition. Worldwide, and estimated about 40% of the crop is lost to the pest and diseases. So you can imagine how productivity we have, what production of the grains and cereals we have. Out of such production, there are 40% losses. And furthermore, the threat of plant pest and the diseases is increased by climate fluctuations. So of course that influences the disease as well as what the pest, agriculture pests are. See, I think this locust, you are very much familiar with this insect, and very recently it was started from very South Africa, and all these are reaching to this India. Of course, there is a problem to control such locust in our country. So of course, and you can imagine how very fast they feed, they are very voracious, and they feed very fast on the entire crop. So they to eat vacated by and the locust and the fall armworm. So all these two examples are very much common for that is very much influenced by the what the changes are. What agriculture we have in India, the total land area about 45.5% of the total land area, total land area is used as agriculture. India is among the top 3 global producers of the major crops, including wheat, rice, cotton, fruits, vegetables, and pulses. And the irrigated crop area of India is 8.2.6 million hectare, which is the largest in the world, and the arable land is 159.7 million hectare, which is second largest world. So of course, such crops are very much influenced by what the seasonal changes, what the variations of temperature, what the rainfall is, all there, that is all together related with such changes. You see what we have faced with this, what changes in the environment is there. This is the data related to 2005. What decline in major crops are there due to this climate change? Wheat we have reduced the 10 to 20%, rice 8 to 12%, maize 5 to 15%, cotton 3 to 10%, pulses about 4 to 10% is there. So of course this is estimated what a loss we have due to this what climate changes are going on. See what demand we have, and if you see by 2025, 2050, we need the what the product we have nowadays that is required double. What the population size increase globally, and accordingly we need to feed the huge population. So of course, one thing is we have to increase the crop yield. Another side we have the climate changes and how this climate change that is affect the what productivity of the crop. As per FAO record, the globe, about 40% of global crop production is lost due to pest. So of course, and this change is because of the what invasive insects are. So with the change in the behavior of the insect, of course, they have shifted from one phase to another phase, like that of one insect is preferring the second stage of the plants are there. So of course nowadays they have been changed into the primary one. So from secondary to primary they have shifted on this, and accordingly they feed on that crops. The major challenges to humankind is a threat to the food security due to emerging and invasive pests. And in India we have recorded 23 invasive alien insects, and out of these, this like that of the papaya millibug, cotton millibug, coconut bites, serpentine leaf miners, tomato leaf miners, all these are few examples related to what invasion of the insects in our countries. If you see the losses we had with regard to the pest, pre-harvest losses are about 40%, and post-harvest what the stored grains are there, and the stored grain that is also consumed by the certain insect. Out of these, 70%, this 70% is post-harvest losses are there. Out of this 10% by insects only. So you can see in your home also you can observe the such insect stored grains like that of the wheat, rice, fruits, all this you can find the appearance of insects are there. So 10% is by this, and 7% by mites, rodents, and diseases are there. Various species of the weevils and the moth that is stored grains like that of the rice, maize weevils, Indian meal moth, all these, and the major affected crops like that of the rice 25 to 41%, maize 20 to 41%, wheat to 28%, and soybeans 11%. So these are the major crops that is being utilized by the ourselves. If you see what pests are there, starting from this weeds to that of the vertebrates. Weeds we have the 30,000 species, fungi, virus, bacteria about 1 lakh. Invertebrates in which the insects, nematodes, and mollusks are about 1,000 species are there, and vertebrates like that of bird, rodents, wild animals, small mammals, more than 272 texts are there. Out of total estimated pest destroying 35 to 40%, crop insects contributing this insects contributing 50 to 20% in India. So what losses we have, and out of these losses, major contribution is due to the insects. See, we have few pictures related to this. This I think this our Western UP is related to sugarcane. So we are working on the white grapes are there. So how this white grape that is affect the roots of the sugarcane. Here you can find the locust, how voracious they are. This Western common rootworm is there. Rootworm is there. See this second other exotic pest. Due to this, what the international trades are there and how these pathogens they reach our country in our country, like that of this, and you can see how the papaya that is infected with the mealybug, this cotton mealybug. You can see this entire plant that is covered with this insect. Again, here you can find the coconut mites. See the picture of this. Of course, they damage the flower in the coconut are there. Tomato leaf binders. If you visit the agriculture field, you will find that the tomato plants that are infected with this, and of course the certain leaf, the lines are white lines you can observe in that. So the productivity that suppressed by these insects. Again, these are few pictures related to this. It is corn leaf aphids, entire leaf that is covered with the insects. Here again, see this white grub. This is very peculiar insect. They move in the agriculture field from upper surface to the lower surface whenever the temperature increases in soil. And see what the practices related to control of this white grub. We generally use the insecticides, what the chemical synthetic pesticides are there, and of course the pesticide that is not able to reach up to that level. So for that there is a requirement of to find out such organisms which can approach these insects. So we are working on this white grubs. So what happens? This when we talk about the climate change and the insect pest. A study indicates that the climate change can alter the activity insect pest, alter the activity of the insect pest. Due to the climate change, there is an increase in the activity of insect pest. It may be in two ways, like that with the rising temperature that boosts the rate at which the insect can digest food, causing them to demolish crop at a faster rate. And second one, this warming temperature causes insect more active and thus more able to reproduce. So whenever population build up, there is more demand of food, and of course in search of the food material, they have to move from one place to another place, like that of the what the locust is there. So this is what this study that indicates related to SCCI, about 50,000 crores value of the crop that is damaged annually in our country. Worldwide, it is 40 billion US dollar are being spent annually on 3 million metric ton of pesticides plus various biological and non-chemical control. So of course, it's how huge amount of insecticides are being used to control this, what notorious insects are there which are damaging our crop. See, insect pest likely to become serious. All these like that of the white flies, fruit flies, mealybugs, thrips, wheat aphids, rice gall, all these are the very serious related to our crop. Certain new emerging major insects like that of the cotton mealybug, white flies, tobacco caterpillars, all these, there is a long list, and of course all such insects they are governed by sort of with what the environmental changes, what the environmental factors are there. So they regulate the population, they regulate the behavior of the insects. There are certain invasive pests, all these coconut bugs, all these, there are a very long list of the insects. Again, the cytosol bites, red mites, all these. And these are few pictures related to the what insects they damaging the product. Like that of this with this mango bud bites, like this all flower that is damaged by the these insects. Here again, lychee bites. This is, and of course, see the product of the what the lychee is there. Again, here you can find the spider bites. Of course, they also damage the crop. Again, what influence the insects? They are influenced by the changes in the environment. Like that of the pests changing from secondary to that of primary, like that of Amicia, this is the Helicobacter armigera, that is the polyphagous, and of course they can feed on various types of the crops, various types of the plants. Leptoptera, Artegonia, aphids, all these, mites, all these. Of course, they have to change their habits from one phase to another phase. So sometimes they don't prefer the primary one or the basic crop over there, but they nowadays they have been changed, shifted from secondary to primary. These are what the pictures related to this insects. Again, Baruca vittata, it is from secondary to primary and changing is there. Recently, and again, see this, the causing up to 42% damage in cow in Andhra Pradesh. So, of course, this is the record. This Andhra Pradesh that is very, very severely infected with this America. Recently, an epidemic of whiteflies was noticed in the cotton growing area of the Haryana and Punjab. So, of course, there is no any localization of a particular insect in a particular area. So, they are moving from one place to another place in search of the food material. So, to control all these, the farmers together with fertilizers and pesticides as a tool to control pests, pest acquiring resistant. This is one very important. Resistant to pesticides leading to the heavy crop loss. Occasionally, farmers are confronted with the pest that can no longer be controlled with the available chemical solution. Because of the insects, they have a very short life period, and of course, they are acquainted with what the environmental factors are there, what the chemical pesticides are being utilized by the farmers, and the cost of the insecticides or the pesticides what being utilized by the farmers, that is very costly affair. So that increase the productivity rate with that. So to minimize to control of all these, we have to search out such organism that can be very much helpful, or that can be called as the farmer's friend, that can be utilized to control this insect pest, and that is very environmental friendly. So with this, we have seen what various types of the insects we have, what productivity that is lost by these insect pests are there, and to control this, that is very important. This when we talk about the entomopathogenic nematodes, I think all of you are familiar with the term of the nematodes. Ascaris, very common. There are so many. There is a long list of the nematodes associated with the animals, associated with the human beings. They are causing so many diseases. But when we talk about such organisms, on this entire earth, there is no any place where there is a nematodes is not found, except this what the Antarctica. Again, say this normal nematode can be that is discovered from the Antarctica, but there is no any place. Just you take any pinch of soil and you can find the nematodes, whether this free living form or the parasitic form or all these. There are different types of nematodes. So when we talk about this entomopathogenic nematodes, they are very much exclusively related with the insects, and there are two categories. One is insect parasitic nematodes, and out of these insect parasitic nematodes, there is one group that is segregated out called as the entomopathogenic nematodes. So they are very much peculiar. They have very much host parasitic relationship, very particular to that of particular species. So of course, we are going to develop such products. They are very much species specific. They are very much target a particular insect, and reaching up to that, they are able to then that they are able to kill the insects. So such groups, the entomopathogenic groups, having the ability to kill the insect pest. How, when, what various practices are there related to this, we will discuss later. Okay. So see this, EPN are insect pathogenic having great potential for biological control, and the beauty of this nematode is they having the association with the bacteria, and of course nematode acting as a vector because of having the movement from one place to another place. They can migrate from one place to another place in search of the insect, and insect also what the soil borne insects are there, they are inside the soil. So of course nematodes, they are able to find out what particular insects are there. So what pathogens are there? Like that, there are two main genera related that having the association with the nematodes and apians, Genoriders and Photoriders, and this Genoriders about what is still today. 31 species have been described from the globe, and Photoriders total 29. Out of these 29, 23 species, new species have been described, and out of this 29, 6 subspecies have been described so far. So when we talk about this subspecies or species, they are very much particular, peculiar to that of a very respected insects. See what from Saharanpur area there is in Uttar Pradesh. Also that is sugar cane belt, and out from this we have isolated some nematodes that having the ability to kill the insect pest within 24 hours. So how fast action of this nematode is there. But in general, the from 24 to 72 hours that is to kill the insect pest. There are two families of the entomopathogenic nematodes, Kashinath and Heterorhabditidae. Out of these two genera, Sternema and Neosternema, are related to this family, and only Heterorhabditis, that is one genera, and very common. Globally, you can find everywhere the presence of such nematodes. What happens? What mechanisms are there? As I have told you that see the nematodes acting as a vector, they have to find out the insect, reaching inside the insect body and releasing the bacteria. Rest of the work that is done by the bacteria. So EPN having symbiotic bacterial association which are released into the insect body. Subsequently, bacteria produce virulence factor like the toxins and the digestive enzymes that suppress the host immune system. So immune system that is suppressed down, and ultimately the insect dies. See, this the presence of the bacteria. This is in case of the Sternema carpophysi, pouch form bacteria that is localized. But see, when we talk about the heterorhabditis, entire gut that is filled with the bacteria. So of the Photoriders that is present in the entire gut in the nematodes, whereas in case of this Sternema, this Genoriders that is very much localized at a particular region. See what happens when the nematodes they get their entry inside the host body. See, this is the free living form of the nematodes. They have to search out the insects for the various larvae present in the soil, reach up to that, and they have to reach either through the what the natural openings are there or by breaking the what the cuticles of the insects. So through that they are able to get their entry inside that, and these nematodes they have to release the bacteria inside the host body, what the insect body is. And from where what the toxins material, what the metabolites are coming out from the bacteria, they have to suppress the immune system. So of course, lots of toxins are produced by the bacteria itself, and ultimately the immune system that is suppressed, and slowly that insect dies. But after that death of this insect, what happens with the nematodes? They have to feed on the what the material is present inside the insect's body, and from where they are able to produce lakhs of nematodes from a single larva. And after death of this, the nematodes they have to come forward from the insect's body. And in terms what the material, depleted material of the insect that can be utilized by the crop or the what the plant is there as a matter. So of course that is the cyclic phases. When we talk about the usefulness of this, they have the broad host range, ability to seek out their host, high virulence resulting in rapid kill, safety to invertebrates and the plants, that can be applied with the other modes of control methods, low cost mass production, prolonged storage that can be stored for a longer time, easy to apply, compatibility with the other control strategy, and EPN are quick, reliable, and environmentally safe. So of course, there is no, still today there is no regard whether that is entomopathogenic nematodes they are affecting the human being or not. Still there is no any regard. Not only human being but also the animal. There is no any regard. See how we are able to identify whether what kind of nematodes we have in by the coloration of the larvae. If see this is the Galeria larvae and infected with the parasites. There you can see the color variation. This is the dark brown color, and if such color variation is observed, there is suspicion of the presence of the heterodactylis. Whereas if creamy color is there, so apparently that can be identified that if such color is there, so the presence of the such nematodes that can be there. See this. This is a single larvae, and out of these single larvae, you can find this sign-like structure. These are nothing but there are lakhs of nematodes are coming out from single larvae. So you can imagine, and how this production, these are female nematodes, and inside the female nematodes, you can find how large number of larvae are present. So after rupturing the water interior region, all these nematodes they can follow from the mother nematodes. So of course this is the structure. Since late 1970s, there has been a tremendous research in commercial interest in EPN and their bacterial associates. What the bacteria? Still we are working on the bacteria related to what the particular strains of the virus are there. So after that, lots of work are going on this. The most up-to-date biogeographical accounts indicates that the EPN have been isolated from all continents except Antarctica. As I have told you that see, when we talk about the entomopathogenic nematodes, means they have to be associated with the insects. And see the presence of the insects, or very less number of insects are there in this region. So still today there is no any record of this EPN from this Antarctica region. From this globe, we have till today 22 species of heterodactyls and 106 species of the Stridina. And out of this, some nematodes they are being utilized to control the insects. So see this, out of this 128 nematodes reported from the globe, till today 13 species have been commercialized globally, including our country also. We are also trying to formulate some products related to the nematodes. This is the list of the nematodes, which are very much peculiar, common, and globally utilized. And based on these about 212 commercial products are being used globally. Out of these 13, various products like that of the medicine, the salt composition is same, but the products are, so many products are there. So like that, globally there are 212 products related to the insects are there. Again, see what these various products are there. And use of these products, cash crop is more utilized. What is the products are used by the cash crop? Relatively slow, like that of tree nurseries, urban grass, turf, and agroforestry and field crops. Very less still today. There is no any such progression in this, despite of what the damage is going on with regard to the insects. See what our Indian scenario we have with this EPM. Initial research in India was conducted primarily with imported exotic species related to what they do. Till today, we have 9 new species that have been reported and globally reported, like that of this Entomopathogenic indica. And see this nematode you can find everywhere on the globe, but it was very much reported from our country. That's why the name of this Entomopathogenic indica that is given. Then Thermophilum, Simai, Megaliensis, Garnai, Stori, Indicum, all these new species have been discovered from our country. All these we have reported from our lab. This is what the Western Uttar Pradesh, and we have isolated such nematodes like that of this Bacteriophora, Pakistanensis, Sopensi, Rho, Siamkai. All these such nematodes we have, and because of we have various isolates, and we are working on this. How this one isolate is very peculiar with that of particular insect to control the particular insect. This is what the species we have, and see what experiments have been conducted by scientists time to time with that of the what particular nematodes are there with regard to the what the various crops in our country. So come to this. This what 96 isolates we have in our lab. We are working on this white grub and Cobarpa and Stolida. These are very, as I have told you that see this white grub problem is a very severe problem in the this sugarcane, and this Helicobarpa and Stolida, they are very polyphagous insects. When we talk about polyphagous means they have to feed on various crops, and that's why. So severity of this Helicobarpa and Stolida is there along with the white grubs. See, when we talk about what the various products are there globally, in abroad, related to Stachynemia carpacapsi, all these such species are very peculiar. There are so many products in different country in abroad we have. Then some products related to the heterophytes, like that of the coffee-4, Darwani, all these, and this is the country where such products are there, and the specificity related to the insects, how they are very much efficacious against such insects. But in our country, we have worked on this Heterophytes indica, Kashinath, Raigo, Kashinath Thermophilum, Kashinath carpacapsi. All these, they are very much tested, and still the scientists they are testing the efficacy of these nematodes on various pests. There are certain products in our country related with what the indicas, carpacapsi, abasi, bicontum, Iro, abasi, all these, and such products are there in the market available in our country also. But see, Other products like that of these green commandos, soil commandos, of course, that were produced in a very ancient time with that previously, and because of such products were there with regard to what the exact nematodes were there, and due to this poor incapability with our system, they were not working like that, and that's why such products were withdrawn from the market. But nowadays, what various products are available in the market, and that can be utilized with the what particular species of the insects are there. So this is the what the company related to this, they are producing such products. See, what scenario we have related to the biopesticides, because day by day there is a need to reduce the chemicals or the synthetic chemical agrochemicals are there, because of they having the very bad effect on the human being directly or indirectly, bad effect on the what the soil fertility is there, or related to the animals. So there is a need to find out to such organisms that can be control the insect pest, what the huge population of the insect pest is there. As report we have in 2024, 2014, biopesticide market value US dollar 1.72 billions, and but 25 global biopesticide market size was valued at 24.2 billion US dollar, and that is projected to reach 97.0 billion dollar by 2036, expanding at compound annual growth rate of 13.5% from 26 to 36. So see this what requirement and what advantages, because of the demand of this, there is a need to produce such enormous amount of the biopesticides, not only in our country but also in abroad. North America dominating the market in 2025 with a 37 to 40% share, and Asia Pacific, India, China is growing rapidly due to rising need for sustainable agriculture, and India expected to be fastest growing market by percentage in 24 to 53. So the wide scope of this nematodes, what the products are there, but there is a need to find out the isolates, peculiar isolates that is very much specific to that of the particular host. So we have a bright future to produce for the production of this EPM-based biofertilizers. All this for the biopesticide market in 2025 we have. See this how acceleration in amount that is there with demand of the what the EPM-based bioproducts are there. See, in our country, what the production we have 1,800 metric tons annually, and this product is by 25-30 firms primarily operating the cottage industry to manage the soil-borne pest. The industry is still considered to be in its infancy. There is no such, because there are lots of agro companies are there which are producing either the fertilizers or all these biopesticides also they producing, but when we talk about this, the EPM-based, the production of the EPM-based biofertilizers, of course, very less companies are there, firms are there, they are producing this. And what demand we have nowadays, that is 24,000 metric tons. This huge amount we require to produce the EPM-based biopesticides. High volume commercial production remains limited. So, of course, there is no such, because see, if we talk about the other European countries and all that, they have certain fermenters, all this, through that fermenters, they are able to produce tons of nematodes, and from that, they are earning lakhs of dollars from that these products. So there is need to come out such agro industries to take over such products and they have to produce in arms. What challenges we have related to this product? Quality control and shelf life. The market faces issues with spurious and unregulated product due to the high demand supply gap. What demand is there and how product is there, of course, there is no such quality control in the country. And shelf life, it is also one important thing because in our country, there is no, because if you see the traders at village level and district level, block levels also, there is no such facilities to keep such biofertilizers because there is a requirement to maintain the life of the organisms also with their efficacy. So of course, this is one problem how we can increase the shelf life of the such products. But in our country, we have about 140 biopesticide production units nationwide, but very few specifically focuses on the EPN products. Very less industry. This is a list few industry related to what the byproducts. So this is what scenario we have. But see what we are doing at our station. We are also facing such problems because how we are able to increase the shelf life, how the quality control of this the nematode can be maintained. Right? So we have developed this small formulation, soil formulation, and nutrient other formulations are there. This is very, and of course certain compositions of such products are there. How we are able to increase because shelf life is very much required to sustain the nematodes for a longer time. So, of course, such products and still we are these products are in the we are trying what the efficacy test is going in the agriculture field because see in lab we have the 100% efficacy of such nematodes are there, but in field, of course, because of certain environmental factors, they have to separate what the activity of the nematode is. Another product is what is the cadaver formulations. Of course, there are such cadavers that can be produced, and it is for the very short period, what the short land area is there that can be applied, and they are very much species specific. One important thing, what we are just talking about the longevity, what the shelf life that can be increased. Of course, this new approach, we have to send the nematodes in the anhydrobiotic conditions because what the thoughts came from from one nematode, there is one entomopathogenic that can be survive up to 35 years without any, if there is a unfavorable environmental conditions. So of course, and whenever the favorable conditions come, of course they have to retain their life cycle. So on the basis of that, we have to send these nematodes half-life, half-dead conditions, and of course, with that can be stored for a longer time. But there is a problem. What survival rate that is very less. So we are again changing what the environmental conditions that is provided for the keeping these nematodes for a longer time. So see this is the live nematodes, and after this what the anhydrobiotic condition, what the condition of the nematodes. So this is new approach, and of course it will take time to reach up to this level to provide such environmental condition and keeping the nematodes for a longer time. And of course that can be available in the market, and because see we know the life cycle pattern of the insects, emergence of the insects are there, and utilization of nematodes accordingly. This is my team who is working on this, whether the taxonomical approach is there or the bacterial approach is there, all this. So any query if you have, you can ask.
Thank you, sir, for your very elaborate research of And it obviously shows why he got the lifetime achievement award. Because at least the first year students who have just completed first sem, they can relate to this. I know arthropoda and platy helminths, nematode. So only examples we have read all these days. But now he has given a very detailed account of all the pictures and all the parasites, how they look like, how the life cycle is going on. Nematodes.
And there is no any hard and fast technique related to this. Just you take out the soil from the agriculture field and
No chemicals, no equipment.
yes, yes.
And you don't need to funding the shots. Because all the challenges are with funding. One is that there is no funding, you can do a lot of research work. That's wonderful that Sir has published 400 papers on this research work.
Thank you, sir, for the excellent and insightful talk, sir. Now I request chair, co-chair, and organizers to kindly present the memento and certificate to our esteemed speaker, Professor Ashok Kumar Chaubey, as a token of our appreciation, sir. Thank you, sir. I also request the speaker and organizers to felicitate our respected co-chair, chair, by the presentees, by the certificates. Thank you, sir. Now it's time for vote of thanks.
Good afternoon, respected Chair, Professor G. Shimta Madam, and esteemed Speaker, Professor Ashok Kumar Chaubey sir, distinguished dignitaries, faculty members, researchers, and dear students. So, good afternoon, one and all. It's my privilege to propose vote of thanks to this invited talk in the session 2. At the outset, I express my sincere and heartfelt gratitude to our distinguished speaker, Professor Ashok Kumar Chaubey sir, for delivering such an insightful and engaging thought-provoking lecture on nematodes. Okay, and they control different types of pathogenic organisms to sustainable agriculture. For delivering and his valuable perspectives and greatly enrich our understanding and will certainly inspire our research in the field. I extend my special thanks to respected Chair, Professor Shamta Madam, for her excellent guidance and for conducting this session effectively. And I would like to thank all the participants for their enthusiastic presence, active interaction during a discussion session. My sincere appreciations goes to organizing committee, faculty members, scholars, students, relatives for their dedicated efforts in ensuring the smooth and successful conduct of this session. So these words, I once again thank everyone for your presence and participation. Thank you all.
Thank you, sir. Good afternoon to all. Respected dignitaries on the dais, of the dais, and research scholars. I hope you are enjoying the sessions. Already 3 sessions completed. After lunch, it will be drowsy. The sessions will be wake up you and enlighten you for your future endeavors. Now the session, we shall begin the session by inviting our esteemed dignitaries onto the dais. I respectfully invite Chair for this session, Professor Bhai Venkaya, a Department of Zoology, Kakti University. Kindly grace the dais. And I request co-chair Dr. Sujata Madam, E. Sujata Madam, Department of Microbiology, Kakti University. And it is my great honor to invite today's talk Professor D. Kashinath sir from NIT Warangal, University. Sir, kindly occupy your chair. Now in this regard, I hand over the session to respect our chair, Professor Venkaya, to kindly proceed and introduce the invited talk speaker, Professor D. Kashinath sir. Thank you, Anand.
Respected sir. Okay, sir. Okay. Respected dignitaries on the dais, esteemed faculty members, distinguished participants and the students. It is my great privilege to introduce the today's invited talk. Professor D. Kashinath sir, Professor in Department of Chemistry at the National Institute of Technology, Warangal. Professor Kashinath sir is a distinguished researcher and academician in the field of organic and medicinal chemistry with the significant contributions to the drug discovery, synthetic methodologies, and green chemistry. He completed his PhD in organic synthesis from the Indian Institute of Technology, Mumbai, and has held prestigious research positions in India and abroad, including the postdoctoral research in France and industrial research experience in the Synergic International Bangalore. With over 3,000 citations and a strong high index, his research has gained wide international recognition. Notably, his review on the Suzuki Myanmar across the coupling of reaction non most highly cited widely. Sir. His research at NIT Warangal focus on the design and synthesis of biologically active molecules, particularly targeting disease such as type 2 diabetes, Alzheimer's disease, epilepsy, neurodegenerative disorders. He has also made significant contributions to the development of green and sustainable synthetic methods, including the catalyst-free reactions and the use of eco-friendly solvents such as water, ionic liquids, and deep aquatic mixers. Professor Kashinath sir has successfully handled several funded research projects from prestigious agencies such as DST and DBT, focusing on novel therapeutic targeting and adverse synthetic strategies. His work bridges fundamental organic chemistry with real world biomedical applications. He has impressive publication record in reputed journals such as Organic Biomolecular Chemistry, New Journals of Chemistry. In addition, his research excellence, Professor Kashinath sir held several important academic and administrative positions, including the head of the Department of Chemistry at NIT Warangal, and continues to contribute actively to academic leadership and curriculum development. With this pleasure, I invite Professor Kashinath sir to deliver your lecture, sir, your talk. Thank you, sir. Thank you.
ఇది అది రికార్డింగ్ ఇందులో ఒకసారి నా బొమ్మ వస్తే సరిపోతుంది ఒక్కసారి వస్తే సరిపోతుంది మాటికి రావాల్సిన అవసరం లేదు Okay. ఏది అది రావాలనే అందులో రావాలి
ఇది కవర్ అయితే సార్
అమ్మ వినబడుతుందా ఇదా సరిగ్గా ఇదేదో స్కాన్ చేసినట్టు ఓకే ఓకే ఫైన్ audible yeah so very good afternoon at the outset I would like to congratulate Istari sir and head of the department and entire zoology department and organizing team for coming up with an excellent program of conducting this 2 days national conference. And also, I would like to thank them for inviting me here to share some of my thoughts related to our research and also in general topic. I would like to mention a special thing here. Actually, I belong to this university. I did my BSc from CKM College, Desai, where I got exposed to the subjects of botany, zoology, and chemistry. I was recollecting my memories of 93, 94, 95, 96, 93 to 96, where we used to come to this campus with friends and we used to tease each other. అరే నువ్వు ఈ క్యాంపస్ రావాలా అంటే నువ్వు ఈ క్యాంపస్ రావాలా అని ఒకరినొకరు టీచ్ చేస్తుంది. Incidentally, all of our friends we got masters and PhDs and settled well. But my memory went to the classrooms of CKM College, where I was taught about the anatomy of plants by Chaubey madam, Leela madam, and taxonomy by Kashinath, microbiology by Ashok, and practice. My humble request is to occupy the front seats, not to occupy last benches, because you will be tagged as a last bencher. Okay, there are so many IAS officers who sat in the last bench, but we are talking about science. That's why to learn science, we should be sitting in the front bench. Okay, so what is this heading? Heading is metabolic pathways. Metabolic pathways. Okay, so what is the meaning of that metabolic pathway? Okay, we see TV, mobile, radio. If we open TV or mobile or radio, there will be so many wires going from one corner to another corner. On micro level, even sometimes you cannot even see with the naked eye, right? But all those wires going from where one end to another end, okay, that going from one end to another end is making, okay, that device to work perfect. That mobile is able to capture wherever I am going. There is a network. Similarly, in our body, there is a network, right? And that network is majorly divided into metabolism of some molecules or metabolic process. Okay, so you have the metabolism complex. Okay, molecules, metabolism of lipids, metabolism of. We are a involves two processes. One is anabolism. Anabolism is making, and catabolism is breaking, right? And without metabolism, we cannot survive. That means without anabolism and catabolism also we cannot survive. A classical example is photosynthesis, making the molecule that is C6H12O6, a solid material, by using the gas that is carbon dioxide and a liquid that is water in the presence of invisible energy that is light in the presence of maybe a pigment. Okay, you are making a solid material. Then the solid material is undergoing polymerization. It is giving polysaccharide, and that polysaccharide, whatever we are eating, that is starch, and that starch is getting degraded into again monosaccharide, again C6H12O6. That monosaccharide is entering into a process called as glycolysis, and the glycolysis is again giving you that carbon dioxide, water, and some energy, right? So the photosynthesis is anabolism and glycolysis is catabolism. Photosynthesis is endo-energy process and glycolysis is exo-energy process. In chemistry, we use two terminologies, that is endothermic and exothermic. Endothermic is consumption and exothermic is giving back. That is what the basis for the biology. That is what the basis for the biology. Maybe I should not talk more biology because you will be throwing so many questions I cannot answer. Right? But to understand the process of drug or to understand the process of a disease, right? At molecular mechanism or molecular level or the mechanism, we should understand what is actually a metabolic process or metabolic pathway and why the metabolic pathway is required to maintain a healthy status. What we say, healthy body, right? No fever, no headache, no body ache, right? No sugar, no diabetes, no cancer, nothing, but healthy body, right? So when I say healthy body, that means all the circuits from one part here, I'm not going to read that, okay? But if you have any abnormality in the complex network, right? That means you are disconnecting from one corner to another corner somewhere, and that disconnection leads to the abnormality at the molecular level of the metabolism, and that is what it is called disease. That is what it is called disease. You might have had nice lunch, maybe chicken, mutton, this kind of might have given mutton or chicken, something like that. Okay, and it was very, very tasty, and I had a little bit more, and in the evening it won't get digested. That means there is something abnormal in my stomach, right? That means either with the food or either with the enzyme or either with the metabolic process where the food has to be digested, it is not undergoing its own natural process. That's why there is a stomach upset. Tomorrow morning there will be a. That's what the disease. In a simple terminology, right? So what do we do? Okay, we talk about so many disease areas, right? So many diseases: cancer, ophthalmology, gynecology, orthopedics, this, that, all therapeutic areas are there, right? Within the therapeutic areas also there are so many diseases. Even if you take cancer, there are multiple diseases, multiple types, right? But even if you consider that, if you look into the molecular level, every disease starts from okay, either breaking a carbon proton bond or carbon oxygen bond or carbon nitrogen bond or carbon sulfur bond or carbon phosphorus bond or maybe something else. Something else means, okay, dissociation of a bond. If you look into chemistry terminology, dissociation of a bond, it may be proton, it may be maybe carbon proton, carbon oxygen, carbon nitrogen, right? So how that dissociation is occurring? What is responsible for the dissociation at the molecular level? Then what is responsible? It is obvious that we use a general term, that is an enzyme, a receptor, right? So we have to understand at that level to understand the mechanism of the disease and what is the byproduct, how the byproduct is produced, what is the organ chemistry mechanism, and how to restore it. That is what actually the drug discovery process, right? That is what actually the drug discovery process. So this network is important, and within that even the therapeutic areas are also important. Maybe I will come little later about the therapeutic areas and all. Okay, but so look into this. We are Indians. We are very, very rich in traditional knowledge. We are very, very rich in traditional knowledge. We have Ayurveda, we have homeopathy, we have Unani, we have allopathy, right? Then can anybody tell what is the difference between Ayurvedic medicine and allopathic medicine? What is, especially the students, what is the difference between an Ayurvedic medicine or Unani medicine or homeopathic medicine and allopathic medicine? Can anybody? I am very happy to get the answer from the students. Okay, fine. So Ayurvedic medicine, we use plant material. Sometimes it is root, sometimes it is maybe stem, it is bark, it is leaf, it is flower, it is seed. Sometimes we use extract. Extract is more used in Unani and also maybe in your what is that? Ayurvedic medicine, whenever you are using, okay, you use the plant material. Allopathic medicine is mostly quantified. What is meant by quantification? You take any ibuprofen tablet or paracetamol tablet. Okay, in the tablet, the drug content is there. If you take ibuprofen 500 milligrams, or 500 milligrams is not the drug. Drug will be some 100 milligrams, and 400 milligrams is additives. Additives which will be helping the drug to be stabilized in that particular formulation, or there may be an additive which will be helping the transportation of the drug particular to its site, or there may be an additive which will be giving to the smell nice fragrance, or there may be an additive which will be giving some flavor so that maybe even the children will be happy to swallow that medicine. Right? So my point here is quantified medicine. That's what the allopathic. That's why we have the dose. That's why maybe doctors prescribe maybe some drugs twice in a day, some drugs twice in a day, some drugs once in a day, some drugs maybe once in 1 week, something like that. Right? There's a reason for that. I will come to that point later. But coming back to this slide, okay, what is that slide is all about, sir? Anything problem, sir? Okay. Okay. मुझे लग रहा है उसको मैं गड़बड़ okay. So our Ayurvedic medicine is known from before Christ. Okay, you look into this one, Ephedra sinica, that is a plant palmahan, and ephedrine is extra power somniferum, opium poppy, it is codeine, betadine, benadryl. This is this molecule actually. Okay, and you have cinchona, quinine based drugs, anti-malarial. Okay, so that is derived from here, and even if you look into the ages, okay, so these plants were used for the treatment of the disease even before the rise. This is the basis for drug discovery for us. Doesn't matter what we do in modern maybe drug discovery programs, but this is the basis. But what is the disadvantage? Disadvantage is we do not know what is the component given in those days. Now we know. But how much amount is that? How much amount is this one present in this plant? That means it is unquantified material we used to consume to treat the disease. Right. I have a second question to the students. Why the Ayurvedic medicine is given over a period of time? Why the Unani medicine is given over a period of time? Why the homeopathic medicine is given over a period of time? Most of the time, Ayurvedic, Unani, and homeopathic medicines are given 2 months, 3 months, 6 months, something like that, but not the allopathic medicine. Because you use plant material in Ayurvedic, plant extract in Unani, and also maybe the homeopathy. When you are using the plant extract or plant material, you do not know how much material is present which is working as a drug. Right? Sometimes you might be taking 100 milligrams of the plant material. In that 100 milligrams of the plant material, there may be only 10 milligrams of the useful drug and 90% of useless drug, and not drug, useless material, which will be entering into the metabolism, which will be entering into the body, which will be entering into the metabolism, and it has to undergo fast metabolism, and it has to be excreted from the body. If it is not excreted from the body, it will be generating something as byproduct, and that byproduct will be leading to the disease, and that disease is new. That's why homeopathic medicine, allopathic, the Ayurvedic medicine, and Unani medicines are given over a period of time, but not the allopathic medicine. Allopathic medicine is quantified. You know how much amount should be given by sex, male, female, by age, infant, adult, by old age, by geography, by ethnicity. The doses are different. Okay, that is the basis for the medicinal chemistry. That is the basis for understanding the drug discovery process. Right. Then, so I'm not going to. I'll skip many slides. I'm going to skip. Okay. Then, yeah, here we know that many. I really appreciate that there are more number of female audiences. Okay. Usually, I dedicate my lecture to female audiences, but today I dedicated my lecture to my BSc teachers. Okay, but now I can come back. Maybe this part of this lecture is dedicated to you all female participants. Okay, so now you see here there is a very interesting molecule. Curcumin. Okay, so this is the structure. Look at the curcumin. How many diseases it is used? Okay, now I have a question to female participants, especially the adults, not youngs. Okay, what is the turmeric powder amount we use in our kitchen per day? What is the amount of turmeric powder we use in our kitchen per day? Huh? Pitch? 5 to 10 grams? 5 to 10 grams. Accept. Okay? Grams. Accept. Okay? So, what our myth, okay, is if you use more turmeric powder in our kitchen, okay, so sometimes we may say that, or we say, not may say, we say that will lead to jaundice. Jaundice. Okay, but do we know how much this curcumin is present in 10 grams of turmeric powder? It is not even 100 milligrams. 100 milligrams. Right? This is the material which is present and useful for so many diseases. Okay, but even if you are using 10 grams, say 1 gram is curcumin and 9 grams is waste material. That means it is entering into our body, right? The waste material and it has to go out of the body, right? That is what it is happening. And my next question is, what is the ideal amount of turmeric powder to be used per person in the kitchen? As per person. Say you are 2 members family, you are 3 members family, you are 4 members family. Okay? Medicinal chemistry literature says that an average healthy human body of 74 kilograms weight can sustain 10 grams of turmeric powder per day. An average weight of 74 kilograms. Okay? Human body with these 74 kilograms of weight can sustain up to 10 grams of turmeric powder. That means that with whatever we are using, okay, if you use more turmeric powder, it will lead to jaundice. It is not true. Now onwards, you can start using more turmeric powder. But try to be balanced. Okay, that is one. Then see here, it is not only disease. It is, in medicinal chemistry term, okay, and this curcumin is used for most of the time for the upregulation. You know that upregulation. Okay, upregulation, downregulation. There are very few where I think this blue one is a downregulation. The blue ones are downregulation, but all yellow color indicated are upregulation of the enzymes, or maybe the metabolic process. That means curcumin can be sustained, and if you use curcumin more, and it will be used for diseases. Then coming to modern drug discovery programs, I don't want to go into the details, okay, but these slides I use for my teaching medicinal chemistry, okay, MSc second year elective. That's why I have some basic definitions also, but I am not going to discuss all those things, but you know these molecules. What are those molecules? Those molecules were made as blockbuster drugs, okay, by starting from the scratch, starting from the understanding of molecular mechanism of the disease to drug discovery, or to bringing them into the market, right? So we use all of them. Maybe I'll come to that later. But in addition to the small molecule, there are very large molecules also in the market. About 25% of the market is based on the natural products, and they are very large molecules, even very difficult to understand their mechanism, but they are blockbuster drugs. Okay, and in addition to that, there is a hybrid type of drug discovery. There is a small molecule and also a natural product. Okay, so you see here, these are all small molecules made in the laboratory, and they were developed by a systematic study of drug discovery program. And these are some of the molecules extracted from the natural products, nature, and converted into drugs. But there are some hybrid molecules, neither this side nor that side. You can still make a molecule and make it to the market and make use as a useful drug by maybe kind of modification, and that modification requires some kind of understanding of molecular mechanism, how that particular molecule is used for that particular disease. Right. Then, yeah, I have some general discussion for the benefit of the audiences, especially students. Okay, why there should be a drug design? Okay, so even if you read the text as it is kept in a simple language. Okay, so to understand, okay, effect of synthetic biological compound on the base of molecular interaction. That's what I was talking about. Process by which it produces the pharmacological effect. You may use a drug. And the drug is not useful. Today we are using mosquito net. Okay, but mosquitoes are not getting died. They are not dying. That means they change their physiology. They change their genetic. Okay, that means they are getting resistant. Right? Like that, sometimes even we may also get resistant. And if you use a particular drug molecule over a period of time, after some time that molecule will not use, especially for the drugs used for the tuberculosis. Okay? Then you have to understand how the drug specifically react with protoplasm and produce particular pharmacology. What is meant by that? Now I have a drug molecule. I have stomach infection. There is some microbial production in the stomach. And I have to kill that selectively without damaging my stomach physiology or without damaging me as a living organism, right? So drug molecule has to be specific to the protoplasm or maybe specific to the tissue or even specific to the organism. That is what to do. Okay, and how they metabolize. Unless you understand the metabolism, it will be very difficult to understand the medicinal chemistry. Okay, one has to understand the metabolism also and the probable relationship. That's what the logic is required to make it run. Right. So that is what a general one and ideal drug. Right. So it should be better than existing molecule, or maybe it should show better selectivity, or maybe good from pharmacokinetics, less toxicity, and also maybe easy removal from the body. Okay. So these are some of the general aspects: how to select a therapeutic area, how to select a drug discovery program, how to select maybe overall process of drug discovery if you are starting with a fresh program. Okay, so first one is you choosing the disease. I want to work on malaria. I want to work on something like maybe cancer. I want to work on diabetes. I want to work on some cardiovascular area. Okay. So choosing the disease, there has to be logic why the disease has to be chosen, right? So even if you consider that point, what are the sub points you have to think about, right? Choosing the disease is accepted, but after disease, what is that? Okay, I want to work on malaria, accepted, no problem. But after that, what you are going to do? Okay, so like that, maybe even if you throw some question, see here, which are presenting prevalent diseases, like the countries. Okay, what is meant by that? I want to work on cancer. I want to make a molecule. I made it. It is working well. I want to release it in Indian market. At the same time, I want to release the molecule in American market. Which will be giving you more money? Obviously, American market will be giving you more money, right? So that point is there. Nobody does the business for loss, right? And impact on the society. You know, during Corona, India produced so many doses and we supply to some other countries. Even today, as a part of corporate social responsibility, many of the companies, okay, part of their revenue, they help poorer countries, especially the African countries, and so much of chloroquine tablets they go to the African countries to treat malaria. That aspect is there, that is mandatory now. Okay. Not only that, even obviously better existing drug economic factors will be there. Nobody does the business for loss. Okay, and not only that, even here also it is very important. There are so many technical terminologies used for drug discovery program. Okay, in that one of the technical terminologies using are maybe agonist and antagonist. This is very important. Okay, whether you want to make agonistic drug for that particular disease or you want to make antagonistic. What is meant by agonistic drug and what is meant by antagonistic drug? Just broadly, I will give you one example. Peptic ulcer is there. Okay, so if there is acidity in the stomach, we take Eno. What is that Eno? Eno produce acid in the stomach and the calcium carbonate will go and neutralize, and that's what we do. But peptic ulcer is something different. Peptic ulcer is overproduction of acid in the stomach. It is because of the H1 receptors. Okay, H1 receptors, and for that you have to inactivate the H1 receptor, and that histidine, cimetidine, ranitidine, Lantac tablet. What is that? Okay, Lantac tablet, what it does, it goes and attacks on the H1 receptor. Okay, and it blocks the activity so that the indigenous molecule produced in the body, that is histidine, it will not interact with the receptor, and that interaction will not lead to the formation of more acid in the stomach. That is what the ranitidine do. Histidine is the biological material produced in the body. It interacts with the H1 receptor, and that interaction leads to the overproduction of HCl in the stomach. That leads to the peptic ulcer. And if you have peptic ulcer, you have to take antacid tablet or maybe antacid drugs. It is not antacid. Okay, the drug which will be working as agonist for the histidine produced in the body. Okay, so it is not histidine, actually it's coming. Okay, so like that, then your molecule will be going and sitting in the same location where your internal molecule is sitting. As a result, you will be controlling the production of acid production in the stomach. That is what the agonistic property. Sorry, the antagonistic property. The agonistic property is, you know, the Parkinson's disease. Parkinson's disease, in the elderly people, they lose the control of the stomach, the neck, and they also lose the control of the limb. That is because of the presence of or absence of, so dopamine in the brain. Absence of the dopamine in the brain, and to treat Parkinson's disease, L-dopa is the drug given. L-dopa, it is given in inactive form. It goes, enters into the brain, and it undergoes metabolism, and it releases the dopamine, and the dopamine is used to treat the. So that means your agonistic or antagonistic property of the drug, when you are starting a new drug discovery program, that is also important to understand. And in addition to that, there are what kind of assay you are going to select. That is also important for a new drug discovery program. This slide is for the new the students. Drug discovery program is broadly divided into two parts. Drug discovery program is broadly divided into two parts. One is pre and second is clinical. In preclinical, a team of scientists, they decide about the disease. They identify initial molecules. Those initial molecules are called as the lead molecules, and the lead molecules will undergo some modification to get you the pharmacophore, and the pharmacophore again used for making number of molecules, and there will be in vitro test. Vitreous in French is glass. Test tube in the laboratory. Okay, so those in vitro tests will be going to in vivo biology and in vivo test to toxicology studies. Okay, so up to here there will be some kind of formulation development and a large production of the molecule. Okay, and by this time company invests a lot of money, so there you have to protect your intellectual property, and the protection of intellectual property at preclinical stage is called as IND. Investigational new drug. IND. Okay, that is very, very important. And once you protect your intellectual property, you can continue the same project, or maybe you want to discontinue, and some others will buy your technology, and they will pay you royalty, and because of the royalty, maybe whatever money you invest up to here, maybe you can reason. Then the next stage is clinical. Clinical study. By this stage, you will have all the information. Information is efficacy. How your molecule is efficacious. Stability. How your molecule is stable in vitro studies and in vivo studies. Toxicity. How your molecule is toxic. How your molecule is behaving at this stage even in the animal. That means you know the pharmacokinetic and pharmacodynamic data of the molecule at this stage. With that data, okay, company will apply to FDA for the permission, and FDA will give you the permission to test on the human volunteers, and that clinical studies is done in 4 phases actually, and first phase they test on healthy human volunteers. In the clinical trials, they test on healthy human volunteers, and in healthy human volunteers, they will not see the efficacy of the drug, but they will see whether the drug is tolerated by a healthy human volunteer or not. Whether the drug is producing any toxicity on healthy human volunteer or not. That is done over a period of maybe 6 months or something like that. The data goes to FDA, and FDA gives you permission to go to phase 2. In phase 2, actual efficacy of the drug will be tested on the patient. On the patient. Okay? So by the molecule enters into the second phase of clinical, you know the toxicity data. With that, FDA gives the permission, and at the second phase, the drug is tested for the efficacy, and toxicity and efficacy studies goes together. And in the third phase, the molecule is tested in larger population. First 2 phases is smaller population, and third phase is larger population and for a longer period of time. First 2 phases is usually for 6 months, 1 year, something like that. But third phase goes for 2 to 3 years for a larger population and different geographical areas. One in India, another in maybe Australia, another maybe South Asia, or maybe Africa, something like that. Different ethnicity. And all the data is collected and all the data is submitted to the FDA, and at the end FDA will give you permission to release the molecule in the market, and even after releasing the molecule in the market also, FDA will have its watch. They will be spying whether your molecule is working well or not. More than FDA, your competitor will be looking for some disadvantage to take. Okay, so that's why it is very, very important to produce the data and document it and produce to the FDA to get clarity on the drug discovery or maybe your molecule. Then, how much overall time takes? Maybe these slides you might have seen so many times because I delivered this lecture so many times. Okay, especially at the network. Okay, so overall, this is in fact old story. Now there are different approaches, but even then also, average drug discovery program goes from current okay is time period is from maybe 7 years to 10 years. In those days it used to take 15 years. It is not only that, drug discovery program is multidisciplinary. You need chemist, you need chemical engineer, you need pharmacologist, you need formulation development, you need medical doctor, you need investor, you need marketing person, you need botanist, you need zoologist, everyone, right? Multiple people are involved and multiple people are paid for maybe 7 to 8 years. That means lot of money is invested. That is what the risky business. That's why one has to be very, very logical. One has to be very, very on point to bring a molecule to the market. Okay. So unless you do logically, then your molecule will not be coming in a shorter period of time. And unless you are bringing the molecule in a shorter period of time, you will not be able to save money for your company. If you are not saving money for your company, you will not sustain. That is what overall picture of modern drug discovery. I am not going to go into the details about all those things, but just after making so many molecules, only one or two molecules comes into the picture. I will show you. Yes. Yeah. So I have defined about the drug, right? Disease. Then for any given target disease, there can be multiple targets. There can be multiple. Maybe I skip all these things. Okay, look into this one. It is not visible. I am sorry. Okay, I did not know that this all is this thing. Otherwise, I would have made a better image. Okay, so even if you take the physiology of a fever, what is fever in layman's language? What is fever? Increase the temperature of the body. Right? You work very hard. There is no rest. There is a body inflammation. And that inflammation leads to the elevated temperatures. There is an injury. There is an inflammation. That inflammation leads to the elevated temperatures. Okay? There is an environmental change that leads to the elevated temperatures. But elevated temperature is a layman's language. But as a chemist, as a biochemist, as a synthetic chemist, as a drug discovery person, what should be my understanding? My understanding should be very molecular. Right? So, if you consider the of fever at a molecular level, even this image image is not, I'm even not, I'm also not readable. It is not visible for me because there is a problem in my eyes. Okay. So, but look into this one. So seriously, there is a problem. Okay. So, actually, prostaglandins are important, and prostaglandins play important role in the elevated temperatures of your body. And these prostaglandins are released in the hypothalamus area of brain. Zoology people, I don't want to talk more about hypothalamus, otherwise you will kill me. Okay. Then you have to control the production of prostaglandins. You have to control the production of prostaglandins. You have to control the metabolism of prostaglandins. Okay, overproduction of prostaglandins will lead to the elevated temperature, and whatever paracetamol tablet you take, okay, through the stomach, it will enter into the brain and it will go to hypothalamus and it will interact with the receptor and it will control the release of prostaglandin or maybe either production or isomerization. Even that isomerization will also lead to the conversion of arachidonic acid into prostaglandin and vice versa. That is the molecular mechanism. Then, if I talk this molecular mechanism to a zoology person, other than botany zoology person, who do not know the physiology, do they understand? No. Okay? That's why, better than to go into the details, I made a general presentation, okay, to not to get scolded by you. Okay? Let us look into some of the examples of okay. Why this is important? ADME. Girls, you might be having some pharmacy students, your friends. Ask them what is ADME. Okay? Why that ADME is so important for a drug discovery? Why that ADME is so important to understand? Okay? We took food. We ate food. Right? Then, just now I was mentioning that food enters into glycolysis and it breaks down into carbon dioxide and water. And I told you that the digestion of the food is exoenergy process. Exoenergy process. Right. That energy is used. Then what purpose that energy is used? That energy is used for making some of the essential metabolites for the body for different functioning, and that energy is used to make my muscle. That energy is used to speak, to throw my voice. That energy is used to move my hand, and that energy is also used to think. Right? Energy is used in multiple ways. That's why that absorbed in multiple ways. Similarly, your drug molecule also, when you are taking the drug molecule, it is absorbed differently in different parts of the body. If you are taking as a tablet, tongue, there are so many microbes, there are so many enzymes which may degrade your molecule. After that, GI tract, stomach, so much of acidity, your drug molecule has to be sustained there, stable. Then intestinal glucose. Intestinal glucose, your drug molecule is entering into the blood. There are multiple enzymes working on the drug molecule which are helping to transport the drug molecule to hypothalamus, to the brain, wherever it is. That is what the distribution, absorption, distribution, right? And also it has to be metabolic processes. It has to show the physiological property. That's why this article is very, very important. And generally, pharmacy people they study more about this. But for us, it is not required. Maybe I'll go to that later. Okay. Then, in fact, every drug is not same. Every drug is not same. Every food is not same. Your tea, coffee is different. Caffeine is different. C6H12O6 is different. Fatty acid is different. A protein, chicken, mutton, it is different. Structurally, it is different. I am organic chemist. I am talking in the context of organic chemistry. Structurally, it is different. Right? Then, it is obviously, when the structure is different, its metabolism will be different. When its metabolism is different, that the endoenergy process is different. When endoenergy process is different, exoenergy and gram-negative bacteria. Right. Then you try to change something, working group, okay, provides steric hindrance. Okay. So this is better. Then even this is better like that. So this is one of the approach. This is one of the approach without like making the molecule without lead structure. Okay. Then this this is a blockbuster. Again, this is one of the top 5 sold drugs in the world today. Okay, tentative name is sildenafil, common commercial name is Viagra. Okay, how it came? It came by accidental discovery. It is not accidental discovery, it came by the observations during the clinical phase. Clinical phase observations are very, very important, and some of you might be knowing that couple of years back in Warangal area, multiple women died. Some company was doing some tests on some gynecological problem, and about 50 women died. Some of you might be knowing. I don't know. Okay, so that's why if some company is asking you to be a volunteer for their drug testing, okay, don't accept answer. Okay, then here this company was working, I think Pfizer. They were working for the diabetes. Okay, so sorry, not diabetes, hypertension. The cardiovascular area, hypertension is the disease, and specific phosphodiesterase inhibition. If you inhibit phosphodiesterase, there will be pulmonary arterial, okay, dilation will be vasodilation, contraction dilation. If you have the dilation properly, your blood flow will be proper. That's how your hypertension can be treated. Okay, so this was the drug target, and during the clinical trials, surprisingly, the company observed the more vasodilation in the male penile part, and the clinical observation was taken as a point to make this molecule as a blockbuster drug, and today this is most sold drug in the world. There are multiple disadvantages, heart attacks, these, that, and all, but still people are using, and even clinical observations, every minute observation is very, very important. Okay, right. Look into this. If you want to start a new drug discovery program, 5 minutes? Okay, no problem. Okay, if you want to start a new drug discovery program, every point is very, very important during preclinical phases. Okay, so see here, this is the progesterone, it is produced in our body. This can be taken as a reference to produce a new molecule, and this was taken as a reference to produce this molecule, not just where this is a partner. Not only that, even this one, endometacin, okay, so serotonin is a molecule, and you know how the vaccines brought within 2-3 months. Forget about this drug discovery. Let us take the latest problem. How the scientists could bring the vaccine within short period of time. Anybody know? No. Okay. Fortunately, before coronavirus, there was a SARS virus. Okay. SARS virus is a protein. COVID virus is a protein. Okay. Fortunately, 90% there was a resemblance between SARS virus and COVID virus. That saved this world today, actually. Okay. So scientists did not worry about the 90%. They worried only 10%. That was studied using genomics, proteomics, and they could bring the vaccine very, very fast. Okay. My point here is, now you don't have to go to some example. Whatever example is around you, you try to take that as an example and try to take that concept as a drug discovery process. No problem. Okay. Like that, maybe you can consider. Metabolic studies are very, very important. Okay. So see here, sildenafil is actual drug given, but this is less active than its metabolite. This is oxidized form, this is reduced form. Prontosil is actual drug, but the sulfonic amide is actual metabolite produced in the body, is working well as like a drug molecule. Okay, like that there are multiple examples, but just this is for the important. Okay, so how the new molecule modification can be done. Okay, so you got so many molecules marketed in the drug. Okay, and out of them, some of you might be knowing that there is a technical term called for pharmacophore. Pharmacophore, to define for the students, pharmacophore is an essential part of the drug molecule to show that particular pharmacological effect. Essential molecule. See, there are number of molecules, but phenyl, and triazole, tetrazole is common for them. So whenever you are taking whenever you are deciding about the pharmacophore, you have to be very, very careful. Once you design the pharmacophore or decide the pharmacophore, you cannot touch that molecule, that part. If you touch that part, okay, it will lead to the something different. You may lose your drug-like property, but the other part can be used, taken as a reference to make your molecule, and all the molecules are blockbuster drugs sold for that particular losartan, that is cardiovascular. Okay, then it is not only that. Whenever you are doing the pharmacophore study, whenever you are doing the modification of the pharmacophore, sometimes it may give you good result, sometimes it may give you bad result. Good result, you take it, no problem. Bad result, you have to record it, you have to submit to FDA. Otherwise, maybe some other company may take up your drug discovery program, and if that bad result, if that company, okay, observes and if you complain to FDA, whatever your invested money for your drug discovery program, it will be in water. Right, this is a classical example. Look into this one, again same benchmark form, okay. So this is actually a molecule, okay, this is the activity, and red part is the pharmacophore. You try to modify the pharmacophore, levorphenol, 4 times active than your original molecule. 4 times active than your original molecule. What is meant by that? If you are taking the benchmark form, morphine, say 100 milligrams, okay, you can take levorphenol only 25 milligrams. Which is better? 100 milligrams is better or 25 milligrams is better? It is obvious that 25 milligrams is better, right? So like that, maybe sometimes you may get better activity, you may lose the activity, okay? And sometimes you may even observe some other activity. Here there is a classical example. Morphine, R and R is a proton here. This is OH, this is also OH. R and R is OH, morphine. R is CS3 here, and R dash is proton, codeine. Why the doctors suggest you not to drive after taking Benadryl cough syrup? Why doctors suggest you not to do so much of activity after taking any cough syrup? Forget about codeine. Okay, so codeine is the part of this molecule, and look into the beauty of organic chemistry. If you change R and R into COCS3, it is getting into heron. You know what is the importance of heron, right? Then, whenever you are doing the medicinal chemistry program, we should be in a position to draw lines. What is that line? Line is, if it is morphine, it is analgesic, and if it is heroin, it will be getting into addictive properties. Right? Okay? Like that, there are multiple approaches for the drug discovery programs, especially for the students. Okay, so whenever sir group is working in what is that, sir? Infectious diseases, right? There was special call by ICMR couple of years back for the infectious diseases, and some of the infectious diseases are also called as orphan diseases, and there was also a special call in orphan diseases. As a student, as a practitioner of science, as a practitioner of medicinal chemistry, as a member or maybe as a person of drug discovery program, we should be having eyes everywhere. Who is doing what? Try to catch it. Take it to your laboratory. Try to do something logically so that you can contribute to the drug discovery program. And if you can make your molecule, like sir is making some of the products into market for the agriculture field, okay, like that, if you can make your molecule into market, and if your molecule is serving the society, what else you want, right? So maybe because of lack of time, I cannot go beyond this, but I have some other concepts also. But maybe for the benefit of the students, I will be very happy to come again. I am a shameless person. I can ask you, I can beg you for gaining knowledge and for sharing knowledge. इस दुनिया में मेरे से ज़्यादा बेकार में आपकी कोई भी नहीं है. Okay? Thank you.
Thank you, sir. Thank you for your lecture. I assure sir, no one wake up. No one gets sleep with your lecture.
I don't know.
Now I request the chair and co-chair to felicitate our Speaker.
Thank you so much, sir, for your very interesting and very elaborative lecture, research-oriented lecture. Then you can utilize this lecture, theme of this lecture for drug discovery. Sir has clearly explained about the drug availability, drug toxicity, clinical trials, entire history. Sir has explained very clearly. Then we know well about the Indian climatic condition is very good for natural vegetation. Number of medicinal plants are available in our environment. We can make use those. They have to extract secondary metabolites, and then they have to discover new type of drugs. Definitely, we know well about the side effects of allopathic drugs. Then definitely we must exploit our natural drugs from medicinal plants and then microorganisms. Okay, from their secondary metabolites. Then I hope that in the near future you may discover new type of drugs from our medicinal plants. Then you can serve the society. Thank you, sir. Thank you, sir, for your very good.
Felicitation.
Felicitation. That's it. It is my privilege to propose vote of thanks for this invited lecture. That is session 3. I extend my sincere thanks and heartfelt gratitude to our distinguished speaker, Professor Kashinath sir, for delivering such an enlightening, informative, and then thought-provoking lecture. His valuable insights have greatly enriched our knowledge and will undoubtedly inspire further research and discussion. And now it is time for felicitation to our speaker. Then one more is we have to remember in Indian knowledge system, actually we are in NEP, in a new education policy, we are introducing Indian knowledge system. That is why definitely we have to discover new type of drugs from our ancient history. We have to understand that. Okay?
Incidentally, I am chairperson for NIT 2020 implementation at NIT Warangal. We are doing so many programs. We will be very happy to maybe conduct some couple of lectures for activities.
Okay. You can make use those lectures. Those lectures will be delivered by sir. Then
I request organizing secretaries, Professor Narayana sir and Professor M.A. sir to grant felicitation to our invite our speaker, Professor D. Kashinath sir. Thank you, sir, for your wonderful lecture. Now I request the speaker, sir, to present our chair, co-chair madam, Dr. Sujata madam, Department of Microbiology. To present the chair.
Munjal Srinivas is my class.
Thank you, madam, for all the questions. Thank you, madam.
Right? Next, అది అక్కడ నుండి తీసేయండి. Actually, one of the invited speakers is waiting from 30 minutes. So he is from Canada. So now the session is.
Good evening. It gives immense pleasure to invite this session. Chair of the session is Chair of the session is Professor E. Narayan sir. Please come on to the dais. It is an online session. Our invited speaker is Venkateshwarlu Kanamla Pudi. He is delivering his lecture from US. Now I am hand over the process to Narayan sir. Please introduce the speakers by Rita.
Thank you. Good afternoon to all.
No, screen. Yeah, you want to.
Okay. It is my great good afternoon to all. Good afternoon, sir. It is my great pleasure to introduce our distinguished invited speaker, Professor Venkateshwarlu Karmarlapudi, a globally recognized expert in molecular and cellular pharmacology. Professor Kanamarlapudi is currently serving as Professor of Molecular and Cellular Pharmacology at Swansea University, United Kingdom. He obtained his foundational education in India, earning his BSc degree from Acharya Nagarjuna University, Andhra Pradesh, followed by MSc in Biochemistry from University of Hyderabad and MTech in Biotechnology from IIT Kharagpur. He later completed his PhD in molecular biology and biotechnology from University of Sheffield and was awarded a prestigious BSc from Swansea University in recognition of his outstanding research contributions. With an